تفضل.
Please, السلام عليكم ورحمة الله. I will connect the phone internet. I will connect the phone internet. It's better.
Kecil.
السلام عليكم ورحمة الله. Do you hear me? Do you hear me? Do you hear me?
Yeah.
Yes. If there is any interruption, please write on the chat box. Please, all of you should mute the microphone so I can speak with you. And anyone who wants to speak, he must get a permission. He must raise his hand. There is a raise of hand, okay? Okay, I'm going to start. I sent you the topics which you are going to study for the written exam and the 2 topics for the practical exam on the WhatsApp. Please shut down your microphone. Mute your microphone because there is a lot of noise. Please, Peter, lock your microphone. Close your microphone. Peter and Gordon Reith. ديسكتوب ايه؟ هي اقفلي دلوقتي، اقفلي دي، افتحي البتاع. Please don't speak and mute your microphone so that I can explain. Please mute your microphone or I'm going to mute all of you. Please mute all your microphone again. كده مقطعها بنعملها شريف دويدار يمكن تكون احسن. مش عارف يتصل ليه.
Doctor, please you mute all the mics.
Okay, I will mute all the mics now, but if I want to ask anyone, I will. Okay. Don't allow. Today, I'm going to speak about glycogen metabolism. If you have studied the glycogen chemistry from carbohydrate chemistry, As you remember, glycogen looks like a branched tree. Okay, I'm going to make from here in order to make to write by the pen. If you look, this is a branched tree. It consists of several glucose molecules connected together by a covalent bond called the glycosidic bond. This is glucose molecule. This is glucose molecule. This is glucose molecule. Several hundred glucose molecule, and this branched tree has several branches, has many hundreds of branches, and each branch is somewhat short, about 8 glucose residue, about 8 glucose molecule connected together in each branch, and this is the chemistry of glycogen. What is the function of glycogen? Glycogen is a store of energy in our body. This is a storage form of glucose in our body. It helps to supply blood glucose during fasting less than 18 hours. Less than 18 hours. If you look, during Ramadan, Muslims fast during Ramadan. They don't eat or drink for about 17 or 18 hours. Blood glucose comes from glycogen. As you know, glucose is essential for the RBCs because the RBCs does not have mitochondria. The only source of energy for the erythrocyte is glucose, and the brain depends only on glucose. So, glycogen supply blood glucose during fasting less than 18 hours. If you are fasting or starving more than 18 hours, the glycogen stores will be depleted. There will be no glycogen in your body. So, glucose comes from a process called the gluconeogenesis. Of course, we are going to discuss gluconeogenesis in a subsequent lecture. Gluconeogenesis means synthesis of glucose from the body proteins or from the body fat. This occurs during starvation for a long period of time, for several days, several months. The function of glycogen. Glycogen supply the muscles only by adenosine triphosphate and energy. Glycogen gives glucose to the muscles only, but muscle does not give blood glucose. Muscle does not supply the body with glucose. Muscle supply itself. I'm going to discuss why the muscle does not give blood glucose, but the liver glycogen is broken and maintain blood glucose. So the function of the liver is to break the glycogen into glucose to supply blood glucose during fasting for a longer period of time. The glycogen constitutes from 8 to 10% of the weight of the liver and constitute about 2% of the weight of the muscles. Glycogen metabolism is divided into glycogenesis, glycogenolysis. Glycogenesis means synthesis of glycogen from glucose to build the glycogen from glucose. Glycogenolysis, breakdown of the glycogen into glucose. I will stop sharing and ask you if you hear me. Is the internet connection is clear? Do you hear me? Do you hear me or do you not hear me?
Yes,
Yes,
Do you hear the previous part of the lecture?
Yes.
Yes, you understand. If anyone does not understand, please write on the chat box.
Doctor.
Please write on the chat box for anything that you do not understand. I'm going to complete. Glycogenesis means the building of glycogen. Building of glycogen or synthesis means to link glucose units together. To link several hundred glucose molecules should be linked together by covalent bond called the glycosidic bond. The building of glycogen or the synthesis of glycogen occur in the liver and muscles. The function to store glucose, a storage form of energy. Of course, for any metabolic pathway, there are key enzymes. The key enzyme of glycogenesis is glycogen synthase and the branching enzyme. But glycogen synthase is the key enzyme. Branching is not the key enzyme. Branching, it helps glycogen synthase. Okay, I'm going to discuss these two items from the figure, from the diagram, because it's much more easier to discuss from the diagram. This is a diagram for the build of the glycogen. The first steps for any metabolism of any carbohydrate or monosaccharide is activation. Chemical activation occurs by a kinase. Kinase is a chemical activator. Activation occurs by binding of phosphoric acid to carbon number 6 of glucose to form glucose 6-phosphate. The kinase gets the phosphoric acid from adenosine triphosphate, convert it into adenosine di, and 1 phosphoric acid bind to glucose to form glucose 6-phosphate. We have studied this reaction in glycolysis. This enzyme is called glucokinase or hexokinase. Another enzyme will function as a mutase. Mutase means it changes the position. Change the position of phosphoric acid from carbon number 6 to carbon number 1. Why I change the position, the location of phosphoric acid? Because the following enzymes act on glucose 1-phosphate. The following enzyme is called uridine diphosphate pyrophosphorylase. Uridine diphosphate glucose. This is glucose. Pyro means 2 phosphoric acid. This enzyme breaks uridine tri. Uridine is uracil. Of course, you know adenine, guanine, cytosine, thymine, uracil. These are nitrogenous bases of nucleic acids. Here there are 3 phosphoric acid. Pyro means 2. Pyro breaks 2 phosphoric acid. PPI means 2 phosphoric acid. And then I will get uridine monophosphate. But here is uridine di. Why it is not mono? Uridine diphosphate glucose. Because glucose, there is a phosphoric acid and glucose 1-phosphate. Then I will add another phosphoric acid from uridine mono. Uridine mono contains 1 phosphoric acid. And glucose 1-phosphate has a second phosphoric acid. So I will get uridine diphosphate. By linking uridine mono to glucose 1-phosphate, you get uridine diphosphate. Why? Why I should sensitize uridine diphosphate glucose? Because uridine diphosphate is the building unit of glycogen. So to build the glycogen, by linking glucose unit together, you must have uridine diphosphate glucose. Of course, the details of the steps are not required from you. The most important is the name of the enzyme, regulation of the key enzyme. The key enzyme, glycogen synthase, this is the key enzyme. Another enzyme which cooperate and help glycogen synthase is the branching enzyme, sensitize glycogen. And the figure will be clear. The next figure. What is this? This is a glycogen primer. I will make a circle around it. What is a primer? Like the base of any building. The primer is a small molecule of glycogen or a small molecule of protein called glycogenin. Glycogenin. This is a protein or a small molecule of glycogen. If you look here, this is 1 glucose unit. This is a second glucose unit. This is a branch point. Glucose, glucose. This is glucose, all of which, but there is a branch point here. Here is a branch point, okay? So several glucose molecules linked together across a branch. This is a primer. How is primer sensitized? You don't have to know in the undergraduate course how primer is sensitized. But you have to know the name of the glycogen synthase enzyme, which is the key enzyme. The key enzyme add glucose. Should it add glucose? No, it add uridine diphosphate glucose. Uridine diphosphate glucose, okay? This is glucose. Add several glucose units. This is glucose unit which is added upon glycogen primer, upon the primer. This is the primer. And this is another glucose molecule added to the primer. A third glucose molecule added and linked by a glycosidic bond. On the other branch, several glucose first, second glucose unit, third glucose unit, 6 glucose unit, 7 glucose units. But we have here one branch. If you look at this structure, there is only one branch. But in glycogen, I have several hundred branches. So a branching enzyme will interfere and create another branch. The branching enzyme will take part, will take this part, will remove this part from the branch point, and then conjugate it to the middle of the adjacent or the neighboring chain. This is a chain. This is a chain. So part from the chain on the left side is removed, and this part is linked to the neighboring chain to create a branch point. So there is a creation of another branch by a branching enzyme. This point of linkage is alpha-1,6 glycosidic linkage. If you study carbohydrate chemistry, I have discussed in the lecture of carbohydrate chemistry what is alpha-1,6 glycosidic bond. So the branching enzyme take a segment from the left branch and add it to the right branch in the middle of the right branch to create a new branching point, okay? As I said before, the primer may be a small segment of glycogen molecule or a protein called the glycogenine. You have to build the glycogen molecule on the glycogen primer. These are the steps which are discussed here. I have discussed it on the diagram. Regulation. The most important thing you have to study is the regulation. I'm going to stop here to ask you if you hear me. Do you hear what I said?
Yes, ma'am.
Yes.
Is everything clear?
Yes.
Yes.
Yes. The internet connection is good or it is interrupted?
It is
It
good.
is good, doctor.
It's
It
good?
is good.
Okay, I'm going to complete. Okay.
Can I ask you a question, doctor?
The most important thing is the regulation of glycogenesis. So, as regard the steps of the synthesis, you don't have to study the steps for the synthesis, but you have to know what is the name of the key enzymes or the two enzymes which synthesize glycogen. This is glycogen synthase and the branching enzyme. But regulation means to control the rate or the speed of glycogenesis. This is for the key enzyme only. The key enzyme is glycogen synthase, okay? So you have to study perfectly by heart the regulation. The regulation occurs by phosphorylation and dephosphorylation. What is the meaning of phosphorylation? Phosphorylation means binding of phosphoric acid to the glycogen synthase, okay? Dephosphorylation means removal of the phosphoric acid from the glycogen synthase. Again, regulation for the glycogen synthase occur by binding or linking phosphoric acid or by removal of the phosphoric acid. So we have to study how phosphoric acid is added or how phosphoric acid is removed. I'm going to explain from the diagram because it's much easier to explain from the diagram. Okay? We will begin by fasting or starvation. By fasting or starvation, anti-insulin hormone arises. If you stop feeding for 2 days or 3 days, stop to take any food, hormones of fasting or starvation increase in the blood. There are multiple hormones, but the most important is glucagon and epinephrine, which is adrenaline. These hormones of starvation or fasting increase the synthesis of cyclic adenosine monophosphate. In the next diagram, I will say why cyclic adenosine monophosphate is elevated. Glucagon, epinephrine increase the level of cyclic adenosine monophosphate. As you see, this is a code, a signal of fasting or starvation. So cyclic adenosine monophosphate is only elevated during fasting or starvation. Okay? Cyclic AMP, the hormone signal or a hormone second messenger, activates a kinase enzyme. What is a kinase? A chemical activator. Chemical activation occurs by binding of phosphoric acid. Why it is protein? Because all enzymes are protein. Why it is capital A? Because I have B and C. Protein kinase A is an enzyme which binds phosphoric acid to glycogen synthase. So enzyme affect another enzyme. Protein kinase A is an enzyme which binds phosphoric acid to the glycogen synthase. What is the glycogen synthase? This is the key enzyme which is responsible for building of the glycogen, for synthesis of the glycogen. If I bind phosphoric acid to glycogen synthase, it will be inactive. Why? This is something strange. Because I said if you bind phosphoric acid to glucose, it will be activated. To fructose, it will be activated. To galactose, it will be activated. If you bind phosphoric acid to any molecule, it will be activated. Why glycogen synthase is inactivated by binding? Because this is not a molecule. This is an enzyme. So some enzymes are activated if you bind phosphoric acid. Other enzymes are inactivated if you bind phosphoric acid. So there is contradiction. Again, glycogen synthase enzyme, which function to build or synthesize glycogen. If you bind phosphoric acid to glycogen synthase, it will become inactive. When does this occur? During starvation. Why in starvation I should inactivate glycogen synthase? Because I don't have glucose. In starvation, this is somebody who stops feeding, prevent feeding for several days, several months. He does not have any blood glucose. How he can build the glycogen? Glycogen is built from excess glucose in the diet, to store excess glucose in the diet. But this is during fasting or starvation. As I say, this is fasting. So I don't have blood glucose, so glycogen synthase should stop function. I don't have to build the glycogen anymore because I don't have blood glucose. Again, during fasting or starvation, anti-insulin hormone is liberated, such as glucagon, epinephrine, which is adrenaline. Glucagon, epinephrine sensitizes cyclic adenosine monophosphate. This is a signal or a code for fasting, and it is a hormone second messenger. Cyclic AMP activate another enzyme, which is kinase. Kinase bind phosphoric acid. Protein kinase is an enzyme which bind phosphoric acid to another enzyme, which is glycogen synthase. If you bind phosphoric acid to glycogen synthase, the scientists give it the letter small b. Glycogen synthase B means inactive. Glycogen synthase A active, small a. So you have I give a stress. If you bind phosphoric acid to glycogen synthase, it will be inactivated. Because we are in fasting or starvation, we don't have blood glucose. On the other hand, if you eat excess carbohydrate, insulin will be liberated. Insulin will break cyclic AMP. I will discuss in the next diagram how insulin breaks cyclic AMP. If cyclic AMP is broken down, the level is decreased, protein kinase A will stop function. Protein kinase A will not function unless cyclic AMP is present. Cyclic AMP is not present if insulin is liberated. After feeding carbohydrate, cyclic AMP level will be low, so protein kinase A will stop function, so glycogen synthase will be active because no phosphoric acid will bind on it. Okay? I will stop here and ask you if you understand. Do you hear me all?
Yes.
Do you hear me?
Yes.
Is there anything unclear or you don't you want to ask about anything?
No.
Everything is clear?
Yes.
Okay,
Yes.
I'm going to complete. If you have any
Yes.
question, please write it in the chat box. If you have any question, write it in the chat box.
Okay.
If you look on the lower part of the diagram, insulin has effect on the lower part of the diagram. Insulin is very powerful. Insulin is the most powerful hormone. Because if someone stop feeding for several days, okay, and then start to eat carbohydrate, insulin will be liberated. Insulin will act on glycogen synthase B and will remove the phosphoric acid from glycogen synthase B by activation of phosphatase A. This is phosphatase A. Phosphatase is an enzyme which hydrolyzes phosphoric acid that is bound to the key enzyme. Glycogen synthase inactive has phosphoric acid, so insulin will interfere to remove the phosphoric acid from glycogen synthase and to convert it into the active form. We are going to stop within 10 minutes, okay? So glycogen synthase will be activated. Please take care of this point. Insulin will have two effects. On the upper part of the diagram, insulin will break cyclic AMP. So protein kinase A will not function. This enzyme will not function. Again, insulin will act on the lower part of the figure to remove the phosphoric acid. It will remove the phosphoric acid from the inactive glycogen synthase. If you remove the phosphoric acid, glycogen synthase will be activated by insulin. This occurs through a phosphatase, which is a hydrolytic enzyme which hydrolyzes phosphoric acid to be broken. Okay? I'm going to get to the second diagram, which is somewhat more complicated. I removed part. If you look here, I removed part of the figure because it will make confusion. Glucagon, adrenaline, epinephrine. If you look during fasting or starvation, glucagon, epinephrine will activate cyclase. What is a cyclase? Cyclase is an enzyme which creates a circle or a cycle from adenosine triphosphate. Glucagon, epinephrine will be liberated during fasting or starvation. Activate a cyclase enzyme called adenyl cyclase. It will sensitize cyclic AMP. As I said before, cyclic AMP is the code of fasting or starvation. It is a hormone second messenger. It is a message to the cells and the organ that we have fasting or starvation. Cyclic AMP will activate protein kinase A. Protein kinase A will bind phosphoric acid on glycogen synthase. So glycogen synthase will be inactive. On the other hand, if insulin is released, if the human body eats carbohydrates, eats excess carbohydrate, insulin will be broken. Insulin will break down cyclic AMP. I will show in another diagram how insulin breaks cyclic AMP. Insulin is a very powerful hormone. Will break the cyclic AMP. This is insulin. If cyclic AMP is broken, protein kinase A will not function. So no phosphoric acid will bind to glycogen synthase. So glycogen synthase will be active. Because of no phosphoric acid, glycogen synthase will be in the active form. Again, insulin act on the lower part of the diagram. Activate a phosphatase enzyme. This is a hydrolytic enzyme which hydrolyzes phosphoric acid, remove the phosphoric acid. So convert the glycogen synthase B from the inactive form to be activated. There is something else here. If I have excess glycogen, I don't need to sensitize any more glycogen. This is called allosteric inhibition of the enzyme. This is called allosteric inhibition. You have studied this in your course of enzymes. I'm going to discuss it when I give you enzymes, okay? If you have excess glucose 6-phosphate, so I have to store the excess glucose in the blood in the form of glycogen, so there will be activation of the inactive enzyme. This is called allosteric activation. Allosteric activation means activation of the inactive enzyme. By excess glucose 6-phosphate, the body have to store the excess glucose 6-phosphate. Okay? I have to stop here and to ask you. I think we are going to get outside the Zoom meeting within 5 minutes, and then we are going to enter again by the same link. Do you hear me?
Yes,
Do you hear me all? I don't hear any one of you.
Okay.
We hear
Please
you,
answer.
Doctor.
You have any
Yeah.
question? Do you have any question? Is everything clear? Do you have any question?
No.
No. Okay, I'm going to complete.
I have
We're
a
going
question.
to
I have
leave
a
the
question.
meeting within 5 minutes. We're going to stop within 5 minutes. This is the action of insulin. Insulin will break cyclic AMP by activation of the enzyme called phosphodiesterase, means 2, esterase means 2 ester bond. Phosphoric acid make a cycle by 2, this is phosphoric acid, by 2 ester bond. Will break the cycle. If I break the 2 ester bond, the cyclic structure will be open, so I will get adenosine monophosphate. It will not be cyclic. If you break the cycle of the diester bond, it will be converted into adenosine monophosphate, which is not cyclic structure. If cyclic AMP is broken, it will stop function. It will not activate the kinase A. It will not activate protein kinase A. So insulin activates the enzyme which breaks cyclic AMP. So insulin decreases the code of fasting. Insulin decreases the code of fasting or starvation by activation of phosphodiesterase. Okay? We're going to shift. I think it's better to begin glycogenolysis in the next session. Glycogenolysis, lysis means breakdown. Breakdown of glycogen. Why I should break down glycogen? To get glucose. When this occurs? During fasting less than 18 hours. During Ramadan, Muslims stops feeding for about 16, 17 or 18 hours. So glycogen is broken to give blood glucose, and this site occurs in the liver and muscle. The key enzyme is glycogen phosphorylase, and there are another enzyme which help glycogen phosphorylase. I'm going to explain from the figure. Okay? Phosphorylase breaks the glucose unit in the outer branch, breaks the glucose unit, break what? Break the bond, the glycosidic bond, by adding phosphoric acid. Phosphorylase means breaking of the chemical bond by adding phosphoric acid. This is the phosphorylase. If you add phosphoric acid to the glucose, it will be converted into glucose 1-phosphate. So the phosphorylase, by adding phosphoric acid, will break a glycosidic bond. And then will break the next glucose by adding phosphoric acid and sensitizes another molecule of glucose 1-phosphate. So phosphorylase will continue to break the glycosidic bond until we have 4 glucose units on either side of a branching point. This is 1, 2, 3, 4, and this is 1, 2, 3, 4. So I have 4 glucose molecules on a branch point. This is a branch point. If you look, this is a branch. Another branching enzyme called glucan transferase will transfer 3 glucose units from one branch to another branch. Will transfer 3 glucose. This is the 3 glucose units which are going to be transferred. It will be transferred from the right branch to be attached to the left branch. These are the 3. So glucan transferase transfer 3 glucose units from the right branch to be attached to the left branch, but not a branching point. It is a straight chain. And 1 glucose unit will be left. This will be removed by a debranching enzyme. Of course, you have to know only the name of the enzyme. You don't have to know this diagram. You have to know the name of the enzyme. Phosphorylase, glucan transferase, and debranching enzyme. Okay? Phosphorylase, by adding phosphoric acid, will create glucose 1-phosphate. And another mutase will interfere. It translocate phosphoric acid from carbon number 1 to carbon number 6. Why I should translocate it from 1 to 6? Glucose 1-phosphate to glucose 6-phosphate, because the next enzyme phosphatase acts only on glucose 6-phosphate. The next enzyme will act on glucose